Good morning, everyone. So, today, our fourth day of this GAN course. Okay, and today we have a first session. Mine. I have a one
Sir, your
talk.
slides are not visible.
Sound is not visible. Sound is not coming.
So sound is okay, but your slide maybe if you're showing
Oh,
it's
okay,
not
okay,
visible.
okay, okay. Oh, yeah, yeah. That I will. I'm doing. And um, so we'll start with our session with my lecture. Then we'll continue with our foreign faculty, the Alicia Bombard, from 2 p.m. So today my talk based on, just to share my slides. Is it visible?
Yes, sir, your screen is visible but not slides yet. Yes, sir, your slides are coming now.
Slider come. Okay. So today my talk is based on the mm, dynamics of DNA and RNA by using the DNA, RNA, and protein complex by using fluorescence probe. Okay. So you know that the DNA, RNA, and the protein complex, that complex is very important mechanism playing in roles in living cells. There are very complicated mechanism in the bacteria, in the human body. So, and this complex mechanism is a very challenging topics in chemistry, physics, biology, to get the understands what how this mechanism is happening. So there are important is that how the dynamics of the DNA, how the dynamics of the protein, and how the dynamics of the only protein, how the dynamics of the protein, DNA, RNA, this triple ternary complex. So everything is very important in context of biology, but only biology you cannot able to solve this complex mechanism. So for this, you need a biophysics physics concept and as well as chemistry concept. Then you can able to solve this complex biological mechanism. So today I will talk with the using the some fluorescent probe, because this complex mechanism, if you do with some other techniques, that means suppose you are doing NMR. Okay, so that is a little bit complicated, because you know NMR, the signals, getting the signals and the analysis of this NMR, this is a little bit complicated process. So how we can able to solve these complicated problems by using a simple techniques, that is called fluorescence technique. That technique is already developed by many scientists and already explained in this GAN course by Alicia Bombarda. And I have also included the fluorescence technique when I have one previous talks, that biophysical techniques for biomolecules. That also I have covered few part. So I will not go here the techniques, whatever the fluorescence technique. So fluorescence, you know that there is a vast range from the spectroscopy to microscopy. So by using this spectroscopy microscopic technique, you can able to get the dynamics of the DNA and RNA, or DNA RNA protein complex. So let's see that what's the DNA and the RNA dynamics of the DNA RNA, or DNA RNA, the protein complex, that we need to look, and how we can able to solve this problem by using the fluorescence spectroscopy or fluorescence microscopy. So, and what probe is required, and how we can approach, and what result we can get, and the different mechanism of this complex biological problems. So I have taken these references from this literature and the books also. So first of all, that nucleic acid actually is described in this our GAN codes, that DNA, especially DNA and G quadruplex. So mm, I'm little bit, because I will describe to you that DNA and RNA and the protein complex. So dynamics of the only DNA, only RNA and the protein complex. So DNA and RNA, this is the nucleic acids and very important molecules in the living species. And that it is already described that DNA is already described, but RNA is not all described. So I will describe also RNA and the DNA both, so that you can will be a clear understanding of this. So you know that in the DNA, that DNA nucleobases is there, and they will form the Watson Crick hydrogen bonding, and they will form a helical structure, and there is a sugar moiety and the phosphate group, and this including all this component will form a helical structure, and that structure is is responsible in many mechanisms in our body. And the RNA, what the sugar is the ribose sugar, but for the DNA is the deoxyribose. And the nucleobases also, there is a difference in the DNA and the RNA. And RNA in general, they are exist in a single stand. You see that, but DNA you see that there is a two stand, and nucleobases are pairing and form a double helix. But in RNA, it will be a single strand, and it will be exist in the cytoplasm. But DNA in genetic nucleus, in the nucleus of the cells, and responsible for this your genetic materials, or your epigenetics informations. That information you can get from this DNA. But RNA also is a very playing important role in making many mechanisms that will come slowly one by one. And it's very important nowadays to know the what's the mechanism, what's the roles of this RNA in the different biological problems. So DNA that have a different types, because of their helicity and the sugar puckering ring, and the different conformation of the nucleobases. That's based on that it is a three types, A, B, and JDNA. JDNA is very zigzag motions, and it's a but BDNA is all stable, and is exists principally. BDNA is majorly exist. So this different type of DNA in exist in different living organisms. So RNA, and that there is also, I told you, that is a very important biological molecules in the living organism. So in RNA, R types are basically the mRNA. Okay, that is the messenger RNA that codes the protein, your protein, that code the mRNA, and the ribosomal RNA and the transfer RNA. That is the non-coding RNA. They will not code the protein, but they are important roles in playing roles in to make the synthesis of the protein and other mechanisms. So it's a you see that how this mRNA, this is the mRNA, you see that this is the nucleobases, and the protein synthesis is happening in the ribosomes. Okay, and the tRNA also playing role. It's bear with the codon, and that codon is responsible for the synthesis of your desired protein. RNA actually the exist in different conformations, and most popular one of the loop is that RNA hairpin. Hairpin. So hairpin is also exist in our in living cells. This you see that there is a hairpin loop, and this is the hairpin stem. Here it will form a the nucleobase form hydrogen bonding, but here you see that there is a form a loop and hydrogen bonding is not there. So it's a RNA actually exists in different structures, different has a hairpin, sometimes a single, so loop structure. So it is very complicated structural informations, and it's still very many things are in regarding the RNA is still unknown. So, and regarding this protein, that protein is the building block of protein for the is the amino acids, and we have 20 essential amino acids, and amino acids are basically the small organic molecules, and that have a an, alpha carbon atom linked to amino group. This is the amino group and the carboxylic group, and um, a hydrogen atom and a variable component in the side chain. And this is side chain. If you vary, then it will be various type of amino acid is there. And amino acid form the peptide bonds, and then it will form a peptide in our systems. And then actually this small peptide and amino acid synthesis. This is the synthesis of the amino acid. That 20 amino acids. You see that how the amino acids are linked with form a peptide in your body mechanism. This is the mRNA and this is the ribosome. Okay, so when um, so in the tRNA come with codon, and that codon will match with the anticodon of mRNA, and that will match. Then that it will form a, but it will carry also one amino acid. That amino acid will come together here, and one another amino acid will come together and form a long polypeptide chain, and then it will form the your desired protein. So you know that there is 20 amino acid is there, and some of the amino acid synthesis in our body, and some of you to take in the diet. And important part for this also in our chromosome, that is also responsible for the gene actually. So in the gene, you know that we have a approximately 20,000 protein coding gene is available in the human living in human body. So in a gene, you see that when the gene is there, that promoter and the exon and intron will be there. But when the splicing mechanism is happening, then exon are removed, and it will form a mature mRNA. That mRNA is required for the synthesis of this your polypeptide or protein. Okay, so playing the gene and gene editing also is an important part in this process, that complex DNA, RNA, and protein complexes. And this, I told you that complex mechanism, that few part is that transcriptions, translations. These are the very one of the important complex mechanism. So transcription. What is transcription? Transcription is the in the DNA, from your DNA template, and it will form the RNA, mRNA. So, and there is a enzyme is called is RNA polymerase enzyme, and that RNA polymerase enzyme will recognize with the DNA template part, and it will form a mRNA chain will grow. And this process is not a simple things. So this process is very complicated, and how this process is happening, that um, actually we need to take help with the fluorescence probe. That suppose you are taken that. You are taking the single molecule freight, or you can take a simple the labeling of this fluorophore into these systems, into the protein in the RNA polymerase protein or DNA. So if you label, and then and how this RNA polymerase when you will recognize in the DNA template, then how this process will happening, and how many the base pair will generate per in real time, you can get the informations for this. So this is a very important the complex mechanism of these transcriptions, and also the translation. Also, translation means RNA. I told you that mRNA to the protein synthesis in the ribosome, and tRNA will also responsible for this. So this is the central dogma in that is called the central dogma, and these two are very complicated mechanism, and need to solve with the help of this biology people. They are also solving this by using their different techniques, and we'll discuss the fluorescence technique, how the fluorescence technique will be helpful to get and to solve this complex structure of these molecules and the mechanism involved into these systems. So protein will form the different structure, that primary structure, secondary structure, tertiary structure, and quaternary structure. So in different structure actually in we need to know the in by using our techniques that will need to understand by uh, different technique is there. You know that CD spectroscopy you can get a secondary structure, and different fluorescence technique also you can get tertiary structure, quaternary structure. So in different particular spectroscopy also responsible to get a different complex structure of the protein. And so if you go to in very in-depth details of the nucleic acids, that DNA and RNA. So in the DNA RNA, I told you that there is a sugar, 5-member sugar ring and phosphate group, and there is a nucleobases. So nucleobases are there in the pyrimidine and the purine base. So pyrimidine base are there in the cytosine, thymine and uracil, and purine base are there adenine and guanine. So, and sugar is deoxyribose sugar and the ribose sugar. The DNA deoxyribose sugar and the RNA is the ribose sugar. Okay, and so when nucleoside means when the base pair, here that the base is there. That means suppose here you have taken that guanine and you replace with the B with the guanine, then it is called guanosine. Okay, but when the phosphatase group is attached, then it is called nucleotide. Okay, one phosphate group, and it's mononucleotide. And the tri, if there's a triphosphate group is there, then it's called trinucleotide. Okay, so so you see that that uracil is the presence in the RNA. Okay, instead of thymine, and other purine base is common for the DNA and RNA both. And so these things you need to very understand, because will will make this particular that nucleotide, sometime nucleoside will make uh, to make a fluorescence that will come slowly, that how we can make this nucleoside and nucleotide as a fluorescence molecule, and would be helpful to get the this complex mechanism of this DNA RNA and the DNA RNA protein complex. So, and you know that when it will form a Watson key brace pair model, that A mm, AU in case of it is in RNA. If it is a DNA, then AT, and GC is common for RNA and DNA. In GC will form a three hydrogen bonding, but AU would form a double two hydrogen bonding. And you see that if you replace with this base here, then you just put it in adenine here. Okay, this part you we need to able to synthesis in some cases, and to know the clear understanding of this complex biological problem. So this is the Watson Crick base pair model. Watson Crick actually crystallized the DNA, and he solved this the structure of the DNA. But sometimes actually Watson Crick base pair model is not obeyed, and then it is form a non-canonical DNA or RNA structure, and it will form a different model is there, the Dobel model, and other the non-canonical the model is exist, and this is not sometimes acceptable in our body, that you need to be repair for this purpose, and that sometimes you can call as a DNA damage. So DNA damage means that whatever preferable conformation, preferable bonding, hydrogen bonding, if it is not the suitable, not matching, then the DNA is not behave like a normal DNA. Then it's a not a Watson Crick model. It should be another non-canonical model, and that will be in the one type of toxicity in your body, and that need to be repaired. We have the proper enzyme that will repair for this non-canonical structure of the DNA or RNA. So I told you that the nucleoside of existing different conformation also, that seen and anti, and the purine and pyrimidine nucleoside also, the both are exist in different conformations, and based on that conformations, their complex structure of this DNA and the RNA is exist, and because of the super buckering and the different the seen anti conformation of the nucleosides of the purine and pyrimidine nucleosides. So and this DNA then when it will form the complex structure that I told you, it is a double helix structure, and it will form in different groups, that major group, minor group, okay, and the so if you want to get the informations for this DNA RNA, so different, suppose any drug molecule you have taken, that drug molecule will bind to the DNA, okay, or suppose any fluorescence molecule you want to get the dynamics of the DNA, you will need to label the fluorescence, or you need to add some fluorescence dye so that it will bind with the DNA. That binding will be the in the major groove, minor groove, or intercalations, alkylations, or it may be the DNA cleavage agents. So whatever you drug, you will be designs that the which mechanism that drug is driven into this DNA, that you need to be know by this structure, that when the groups binding, then it splits to the sometimes minor group, major group intercalation, then it will be the sandwich with the two base pairs, and sometimes it will be formed by alkylations and cleavage. I told you that it's break the DNA chain by oxidative cleavage, and it will damage the DNA. So different purposes there. Suppose that you have some disease there, you need to be have destroy the DNA. You need to be add some DNA cleavage agent so that you can destroy the DNA by um, by cleaving the DNA strand. So for all of it are the important the parts of this the DNA cleavage mechanism. So mechanism is a oxidative cleavage, and the different drug is already actually developed to, suppose you have a some cancer disease, and cancer particular one drug is some doxorubicin or something, one drug is there. That drug, how the it happened with the DNA. So that mechanism you need to understand very clearly, that how drug is binding with this different special position in the DNA. So DNA and RNA, I told you that you can synthesize the short form of this part, that is called oligonucleotide. Okay, that you can synthesis. There is a pharmaceutical company. They can. Uh, there is a lot much process. There is a process is there, and by using this process, you can synthesize the a short part of the DNA, any sequence part of the DNA. So, mm, uh, so you see that suppose you want to synthesis that deoxy that CGTA. So you did see that this is the cytosine, and this is the guanine, and this is the thymine and adenine. So this part, this chain, this short chain, this chain you can able to synthesis. So one end is 5 prime hydroxyl group, okay, and other end you see that 3 prime end. This is also the hydroxyl group. So these two end, if you finish, then it will go a one chain. You see that here OH is there, here OH is there. That chain you can synthesis in the oligosynthesizer machines, okay. And whatever that short part you want to take in your research, that you can able to synthesis in the oligosynthesizer machines. So so this is the single strand, and another strand you can also synthesize to make a double strand. And so for this RNA also you can able to synthesize one particular sequence. Suppose you want a 12 mat or something, 12 nucleotide sequence you want to synthesize. So 12 nucleobases you need to add one by one each steps, and there is a cycles, and that cycles it will form and to synthesize the sequence. So this is the synthesis methods. You see that how I told you that synthesis of this particular sequence you can able to do. Okay. So first of all, that CPG in that one that solid support that will be attached with the DMT protected one nucleosides. Okay. And there is a steps is that first steps is called detrilations. DMT is out from there, and then this deprotected of the DMT and will couple with the one phosphoramide of this another nucleobases, and then these two then will form a coupling, and activation and a coupling will happen. Then these two nucleoside you see that they will couple. So chain is growing, and here that whatever you are adding here, that is called phosphoramide and DMT protected phosphoramide, and DMT is still here, is still there. So then you have to be some capping agent, okay. Capping is so that the process will be the effective and yield will be the very high. And then the fourth step is called oxidations. You need to oxidize and oxidize the this phosphate group, and need to form a proper and the phosphate residue present in the DNA. And next cycle, same thing. Next cycle, you will add another nucleic acid, and the chain will grow like this way. Okay, so chain will grow like this way, and you can able to when you clip this the solid support, then you will get your desired chain. So according to that, you can synthesize the sequence. I told you that suppose you want to synthesize CGTA, suppose you want to synthesize GGG. So as you desire, you can able to synthesize your sequence, okay. So it is a very and lot of pharmaceutical company actually based on simple this idea, and they're making and producing lot of nucleotide and giving to the institute academia and in different research labs. So the research is going smoothly and so on. And few lab also that they are already have developed these techniques, and they are synthesizing this sequence in the lab also. So this whatever that sequence is developed, that developed sequence will use in the our purpose. And suppose you want to label some fluorescence dye, here also you can attach your desired dye or desired that your probe, fluorescence probe, that I will come that how. So suppose that here that whatever nucleobase is there, that nucleobase if you replace with a some fluorescence dye, then you can make a that chain is a fluorescence, okay. Then you can get the desired information, that information you want to take, Suppose this short peptide you want to apply in the different mechanism. That mechanism you want to track, and by using the fluorescence probe, you can easily able to track this mm, by using this fluorescent nucleotide or nucleoside on that or a chain for this long chain which is level with the particular positions the fluorescence. Suppose you want to place with a suppose this position, or suppose you want to place with a 8 9th position. So this is a long chain is there. So you according to that you can label the fluorescence probe different positions. If you want to level with a probe which is a terminal, you want to level with mm, the in between the two bases. So as you desire, you can able to make the chain of this DNA and RNA and make it a fluorescence with a and fluorescence probe level with a fluorescence probe by using this solid phase synthesis. So now I will come to the that fluorescence probe because you need to help with that take the fluorescence probe so that you can easily solve this the complex biological problems. So fluorescence probe are intrinsic fluorescence probe is that in protein is that tryptophan, tyrosine, and phenylalanine, and naturally there is a NADH and FAD. Then these two are also that fluorescence, and there is also exist in our in the living organisms. So so protein, you know that fluorescence is mainly responsible for this tryptophan moiety, and by monitoring the tryptophan, you can get the fluorescence analytical informations from the protein. But sometimes you know that it depends on the tryptophan extent, how much tryptophan is present, and what the position of the tryptophan is there, so that you can monitor and you can get the because tryptophan only quantum is very low. So suppose you want to get some complex mechanism. So suppose three protein, RNA, DNA, and the protein complex is there. So these complicated structures sometimes you maybe not get a proper signal from the tryptophan protein in the protein tryptophan presence, and how much I told you the tryptophan. Suppose one tryptophan present, so then it is not desirable that you can get a proper signals from this or only one tryptophan in the protein. So it depends on the protein and the situations and the mechanism how it's happening in the suppose in the living cells. In the living cells, the the extent of this fluorescence from the tryptophan is very low that you cannot able to solve. And the image, suppose you want to image, that image also is not possible by using only simple tryptophan. So so these are the natural and intrinsic the species that is present in our living in the living systems that will be the useful sometimes, and but in a very complicated systems that it's not useful because of very I told you that quantum yield and it's is very low, that will not suitable for this and for the to understand the complex mechanism. So you see that that tryptophan and the fluorescence, but phenylalanine fluorescence is very low, and this is the tyrosine the fluorescence, and I know the NADH have a also fluorescence, NADH and FAD also fluorescence. And then you see that this NADH is the absorptions around 350, and here it's fluorescence around 480 something. So FAD also that is as a particular absorption fluorescence. And, but I told you that this is may not sometimes sufficient to get a particular mechanism if you want to understand the particular mechanism. But to understand the basics fundamental of this protein and the basic fundamental of this different enzyme cofactor, that is that you can able to get in vitro, but in not in living cells, that you can understand in the laboratory what's happening the different that enzyme cofactor, what is the role of this NADH in the different redox mechanism that you can use by using this intensive fluorescence probe. So extensive fluorescence probe that I told you that you that that is the that fluorescence probe that you have used externally, so that you can get the your desired analysis. So DNA and the lipid that is not the intrinsic fluorescence, does not show any fluorescence. So for this you need to be use the labeling for this, or you need to be to make the DNA stand or the lipid any to label or to attach something, or mm, to make the fluorescence, so that you can get the informations of this DNA, how DNA dynamics and DNA protein complex informations. So, and you see that fluorescence is obtained by the labeling of the molecule with extrinsic probe. Okay, for protein, it is frequently desirable to label them with chromophore with longer excitation and emissions wavelength than the aromatic residue. So in the protein, you sometimes you need to label. I told you that because of the limitation of the quantum unit and so on, and other factors, other positions, and different environment into this diptropine and so on. So sometimes you need to label the proteins with a desirable some fluorophore, but it should be so that you need to label that should be longer than the acceleration of this the intrinsic probe present in the protein. So this is the very three are important, that Psi 3, Psi 5, and Psi 7 are very important extensive probe that is used in protein and even in DNA. So they have a reasonable the absorption and emission peaks, which is very far from the intrinsic that probe present in the protein, that is used in single molecule freight and FCAs and so on. So FCAs I have already discussed that it is a fluorescence, correlations, and spectroscopy. And FCAs is used in different in the different in, to know understand the dynamics of this the by macromolecules, the protein DNA and protein DNA RNA complex also. So protein labeling that these dyes are also used for protein labeling. You see that DNAs and FITC, okay, and acrylodone. So these dyes vary from stable link labeling with the protein, and to probe the protein to get the information. I told you that protein dynamics in the living cells, protein dynamics in a particular mm, in vitro study, in vivo study you can do by using this. And nowadays also protein you can label with a different tag also, you can do protein tag also, particular tagging by using a different fluorescence protein. So you see here also there is one important dye, it's called ANAs. Even ANAs actually would be a it will form a strong binding, and it will keep the fluorescence emissions very reasonable, so that you can able to track that the BSA molecules dynamics by using the ANAs. You see that this is the BSA emissions and this is the ANAs emissions. So that you see that image emissions is forming stable complex, and you will get the high fluorescence with the ANS and the BSA complex. And you see that the HSA and the ANS, if you mix them, you see that very high intensity of the fluorescence, and which determine that a strong binding of the ANS with the sorry, HSA. So you can using this ANS, so how the HS dynamics and in cells in living organism in vitro you can easily determine by using this probe. So another important that fluorescence and the protein, this is also used to to track the protein dynamics in the different the living cells also. You can get image also. So some fluorophore actually you cannot able to image because of the bleaching, photo bleaching, quantum Hill, factor of quantum Hill. But fluorescence protein is very useful to get the image in the living cells also. And there are different the fluorescence probe GFP is already developed, EGFP. So these are the very important, and they will form a cyclic structure of this COV4, which is responsible for this highly fluorescence of this green fluorescent protein. And that protein is very much useful for get on the protein imaging in the cells, and also the protein dynamics in the in vivo and in vitro. In vitro. So DNA probe. This is the all this discussed type the protein probe, and the DNA probe. You see that one of the most important DNA probe, that's the Ethereum bromide, and Ethereum bromide is very weak fluorescence in water, but when it will bind to the DNA, then its fluorescence very high, 30-fold increase of the fluorescence to the DNA. Okay. And the lifetime also, that Ethereum bromide is 1.7 nanosecond in water, but when it will bind to the DNA, the Ethereum bromide, strong binding with the DNA, and then it will form the 20 nanoseconds lifetime to the double helix DNA. Acridine orange, this is also a very useful integrating probe in the DNA that will be used to get the DNA ladder image. When you do some gel also, that people are using these dyes so to get the image of the DNA. And also in the fluorescence spectroscopy, microscopy, people are using those dyes and to get the information of the DNA and its dynamics. So, other that minor groove DNA binding, the probe is DAPI and Hodges. That these are also it's very important dye that to get the nucleus image. That in the suppose you see that in the this one cells, that cells nucleus, this is a DAPI level. This is DAPI level. So DAPI, if you level this DAPI, you can easily to get the nucleus image in the cells. Okay, so that's why this dye is very still now is very important, playing role in the chromatin dynamics, chromatin informations, imaging in the single molecules, in the ensemble. So all this informations, all this in this technical information, you can use this dyes for to get the image of this nucleus of this in a particular cells. So still now I'm talking with that the fluorescence dye, that is organic dye, that cyanide, PAM. I told you that the DAPI. So this all these dyes, if you level, you can level. I told you that that cycle I have shown that how the oligonucleotide are the forming, and during these formations you can, I told you that you can level in different positions in the terminal internal leveling you can do. So different leveling you can do in the particular that positions. So this dye, you see that internal leveling here, that in the base nucleobases it is level with the fluorescence dye. So you can level in the phosphate here in the group also internal leveling. So according to your discernment, you can level the fluorescence dye into this nucleic acid structure. So, but there is a limitation that if you label those dyes, actually those dyes are very big size and so on. So there is a perturbations and deformation of the nucleic structure and the stability, and that, and also you will miss. Suppose one protein is interacting with this DNA, then the whatever interaction, that interaction you may miss the informations because of this large dye present in the nucleic acids, particular their positions. So for this, for the to tackle these problems, so important is that DNA base analog to develop the DNA base analog. So one of the important base analog that popular is that 2AP 2-aminopurine. Okay, that is extensively used in the research because of this is fluorescence. and is a very small molecule, an ideal structure that uh, you can get the informations mm, from the DNA, okay, and in the RNA to get the dynamics in vitro. And it's depend on the quantum yield. 2AP quantum yield is very low. That's why in the cells is very challenging to get the data from the 2AP. But we have actually developed different probes developed. We have developed thionoguanosine. This is the guanosine analogs. This is the adenine. 2AP is the adenine analog, and thionoguanosine, you see that this is the guanosine analog. And also we have developed the TC nitro. This is cytosine analog. This is also very highly fluorescent molecule, and this is the act as a Fred probe actually. One is act as a donor, and another is act as a acceptor. So if you use this TC nitro, thionoguanosine, 2AP, so there is a no perturbation in the nucleic acid structure, and you will get your desired informations. And this is the latest advanced technique, because if you use the conventional organic dye and other molecules, there is a that limitations. Actually, due to this limitation, you will miss your desired informations. So 2AP, you see that. So suppose 2AP when labeled into this helical structure, when there is a strong stacking, then there is a you see that low fluorescence. But if it is flexible and is dynamic very, then you will get high fluorescence because of the due to the stacking. There is a high electron transfer is happening from the 2AP to G, which is quench the 2AP fluorescence. So that's a one disadvantage of the 2AP. So so this is the use that you can use the particular, suppose this position you replace with the 2AP with the adenine, and then you can get your desired informations. So similarly, that this is the fluorescence, all the fluorescence alphabet. Alphabet means that A, G, this is the analog, A analog, G analog, U analog, C analog. So if you this analog if you use, and then you will make a emissive RNA structures, because all analogs are, these are all the fluorescence, and it is the very isomorphic analogs of the RNA nucleobases. And this is also used extensively in different structural determination of this RNA and the RNA dynamics. So I will explain a little bit that thionoguanosine. You see that thionoguanosine is formed in different tautomer. These two tautomer, one is H1 and H3, and that we have characterized using different spectroscopic techniques. And this H1, H1, these two tautomer have very high fluorescence, and their quantum field is very high. Lifetime, long lived lifetime, they are existing the long lived lifetime. So if you THG and 2EP, if you compare, then you can see that if suppose this is a one DNA stand you have taken, and that DNA stands you this particular position you replace with the 2EP, or you will replace with the THG. So in case of THG, you see that fluorescence is reasonable, very high even in the duplex. But when you use the 2EP, 2EP, that it is a free dye, free nucleosides. But when you level into this duplex structure, you see the how quenching is happening. Is a strong quenching happening in the duplex structure. That's the advantage. But THG, it's not quench so much, so you can use the THC and you can get the desired informations. And also the 2EP have a multiple the lifetime component that is sometimes very tough to predict the what happening into this the mechanism that any DNA that's DNA molecules how they interact with the protein, that mechanism if you want to predict with by using the lifetime, that is also very tough because of the its multiple decay component. But the THG have a specific decay component, so it's a mono or di exponential component. So that component is very easy to predict the structure of the THG, the structure of the DNA molecule by using the THG. So THC, so this is the one work done. So you see the THC is labeled with the particular positions, and then it will varying the DNA basis. DNA basis varies with the with its neighboring DNA basis, and its opposite nucleobases. It varies when it's opposite to the cytosine, that is called match sequence, because THC is guanosine analog. So when it is not cytosine, then it is called mismatch sequence. So match mismatch sequence, you can easily also get the informations by using the THC, and so you can take a particular model for HIV model. Suppose HIV 18 one particular model. In the model, you have labeled the THC at the particular positions, and then you want to get the idea that how this this that that THG level stand was playing the roles uh, in the uh, in the living cells, or how the mechanism is happening, that you can guess easily by using this fluorescence technique, because THG high fluorescence and its quantum is very high. Lifetime, you will, from the lifetime decay component, you can also predict that how its dynamics is modulated. So, fluorescence decay we did, and we have characterized the fluorescence in different flanking bases, and also the its opposite bases, so that you can get that match mismatch sequence as all always, and also the molecular dynamic simulations also we have used, and to correlate with the experiment. And it's very, you see that molecular dynamic simulation when TG opposite to G, then that TG is expelled from the duplex stand. That also we have confirmed using the lifetime also, that we have confirmed that you will get the THG fluorescence when it is expelled from the duplex stand. You will get more solvent exposed the THG lifetime, and that we have also characterized in the fluorescence spectroscopy, and as well as in the theoretical study. So this is the one empty simulation study also. This is a THG. You see the THG is labeled here, okay, in a duplex stand, and different planking and opposite ways we have taken, and how this THG is expelled from the stand, and it's moving from the stands orientations that we get from this empty simulations data. We have checked also the melting curve, and the stability. We have checked that different if you change this the opposite and then the flanking basis, that how the stability of the stand is varies with this TG level, the stand, that we have also checked, and the thermodynamic stability we have calculated from there, and we can get that how where what's the mean the positions that, or what's the conditions that we can the THG is level into particular DNA stand, particular positions. So I told you that the mechanism for the DNA and in our living systems is a very complicated. The complicated is transcription is a very complicated mechanism. Translation is very much complicated mechanism. And there are like this, there are many mechanisms are very much complicated and responsible in very particular, very important roles to understand this mechanism. So one of the important mechanisms is DNA methylations. DNA methylation, this is also is responsible for this epigenetics. Suppose two person is not same. So two, if you take two cat, you see that it's not exactly same. Two dog, if you compare, then it's not exactly same. Sometimes twin is coming. That's different thing. But this epigenetics, that epigenetics is you can mm, this is a very important roles in the in our species generations, and the differentiate one to each other. Because of this, DNA methylation was is important one of the roles playing the roles. So DNA methylations is the methylation of the cytosine in the CPG island. So there is a protein is involved into this DNA methylations. So SRA one domain, this SRA domain of UHRF, there is also UHRF on protein is responsible. So SRA domain when it will bind with this heavy methylated CpG island, then the methylation is happening by using the UHRF and DMT recruitment. So both two are protein is responsible, and DMT is recruited when the UHRF will bind and form the methylation of the cytosine. So due to this very important part, and during this methylation, this flipping of the methylated cytosine happening into the CpG island, and that mechanism, that complex mechanism, is we need to track that how this mechanism is happening. Suppose sometimes we need to inhibit these mechanisms, and then how you can inhibit, what inhibitor you can use based on that. So this is the important information, so that you can take the DNA methylation also playing the role in the cancer. So many diseases are happening in a link with this complex biological mechanism, and you need to understand, we need to understand how this mechanism is happening, that flipping is happening, what protein is responsible, and in what extent. So for this, by using simple fluorescent probe, you can able to get the idea. So you see that this is the work that they have taken the heavy methylated cytosine and methylated cytosine, and they have used that the UHRF and DMT, that when they will bind with that, and you can get an equilibrium constant, and the clipping of these molecules, when it clip, then you can get a high diffusions, and then this stands actually is level with the THG. You understand this? So due to this TH, you are getting the fluorescence, and when the clipping is happening, then you will get a high fluorescence. And these things, not only fluorescence, it is also is by using the stop flow kinetics, and the theoretically also it's proven that flipping is happening, base flipping is happening when the SRA domain of the UCRF is bind, and you will get complete understanding that DNA methylations in the process by using simple fluorescence technique, and using the simple THG as a labeling nucleobase, fluorescence nucleobase. So you know that CRISPR also is a very important playing roles in the ternary complex. I'm discussing that DNA, RNA, the ternary complex. Okay, so here it is the DNA RNA ternary complex. DNA methylations is a protein and DNA one complex, but ternary complex, the DNA RNA protein. So three are in a form a complex. This is a very tough and complex mechanism. So that CRISPR Cas is a cleavage of the genomic DNA, double-stranded DNA, by using that RNA and Cas9 cleavage enzyme. So this is the your target, that you want to cleave the DNA, and this is the RNA, that single guide RNA. That guide RNA when match with the target sequence, then the Cas9 will form a complex, and when form a complex, then it's a nuclease activity. Due to this nuclear activity, then it will cleave the DNA with a double stranded. So this is the modern technique, and CRISPR got also Nobel Prize in 2020 in chemistry, and you know that nowadays that CRISPR gene editing is a very important and playing roles in different diseases also. Suppose you have a disease, a particular disease, and you want to edit the gene, whatever gene is responsible that you want to edit, that using simple CRISPR-Cas technique, you can able to edit that particular gene into this your desired gene for this. So for this actually, you see that I told you that you need a RNA molecules. However, RNA I have discussed that RNA is form a very complex structure. You see that single guide RNA, it will form a very complex structure. The stem loop is there, and it's a long RNA, and it's very complex and dynamic property. So if you did any mutation or any something, that complex formation is may not form in that case, and you will not get that your desired double-stranded break. So by using this CRISPR, that not only simple just editing of this DNA, but also you can do in the different the gene editing, and so that you can make in a different positions, you can edit, you modify, you can mutate mutations, so many things in the biology people are using, and the biophysics people are using this technique, so that they are taking that into the real time by using the fluorescence molecules, what happening in the body or in the living cells, that mechanism, genomic loci mechanism in the gene, and how the double-stranded DNA breaking is happening, and so on, by using the fluorescence technique, by using single molecule freight, FCAs, you can able to do this thing. So 2AP also is used here for this CRISPR-Cas gene editing systems, and other nucleobases you can able to use also for these systems. So single guide RNA, you see that single guide RNA very complex structure. So it's a 20 mer. I told you this is a guide sequence, and you see that this is the CR repeat, and this is a trans RNA, and you see that there is a loop, and two loop is there. One, two, three, three loop is there. Steam loop there. So each part is very important to form in the complex of this structure, and you see that how this complex structure of this is the target, and this is anti-repeat, and this is the sgRNA, single guide RNA, and that is very complicated and dynamic structure of this RNA, and playing very important role for this gene editing systems. And this is actually generally it is in a bacterial defense system. Actually, bacteria is do the defense for this purpose. But this defense system you can use in the different organism also, and people are trying to apply this mechanism in the human also, and to get the to edit the desired gene in your body. So here you see that D DCAS9 and EGFP, they are couple, okay, and single guide RNA taken, and that you, if you deliver it into these cells, then you can get the image, genomic loci image, that um, uh, how the editing is happening, and you can get it that particularly that cell programming, how the cell is programming into this systems, edit the your desired gene, whatever gene you are edit based on the target, you need to design the sgRNA, and you can get the genomic loci image to visualize the chromatin and its mm, gene editing the process by using this fluorescence probe. So by using single molecule, if you use the freight probe, also you can get the spatiotemporal information of this complex mechanism. So this is a very complex mechanism where the DNA, RNA, and the protein, three are formed and forming a ternary complex, and will give a complex mechanism. So okay, I think I will stop here. So you see that how the fluorescence is very important, playing roles in different mechanism. Okay, and this is the very important part in the mm, in the biological research, and as well as that this complex, to solve this complex mechanism, you need to help with the chemist and the physics also.
Okay, thank you so much, sir, for such an informative talk. If anybody wants to ask something, please go ahead. Uh, so I think no questions. Okay, sir, thank you so much. And I hope you all found this lecture very informative. So we will again continue this seminar and we will meet again for our next talk at 2 p.m. by Dr. Aliza. Thank you.
Okay, thank you everyone.